not that easy to remember. So the game-changing drugs such as declatazvir, a drug for hepatitis C virus, Ridiplam, a drug for spinal muscular atrophy, or Lumacaftor, a drug for cystic fibrosis, have been discovered thanks to phenotypic drug discovery. And now the question to you, how many of you have experience with that kind of approach of drug discovery? Please raise your hand. Okay, so not many of you, that's great. So today's panel, innovative approaches to phenotypic drug discovery, will give a chance to those that have never had an opportunity to get a deeper into the topic, but also will shed a light on innovative approaches for those that already have expertise. We hope that also there will be a space to find new ideas for research. So my name is Kaja Milanska-Zabel, and I am the Business Development Executive and Board Member at Ardigen. Today, I have the pleasure to be your host for this panel, and it is my great honor to introduce to you our keynote speaker, Professor Danuta Mosakowska-Earnshaw, Director of Małopolska Center of Biotechnology and a professor at the Jagiellonian University. Professor has gained extensive experience in leading and managing science, from being a biology leader in an international team to head of the biological reagent and assay development team at GlaxoSmithKline. Over the past 7 years at GSK, she has led industrial and academic partnerships in Europe and Australia, developing a passion for supporting academics and turning their research into reality. Please warmly welcome Professor Danuta Mosakowska-Earnshaw.
Thank you for that. Right. So, I'm also dedicate a little bit of my time as a Royal Society entrepreneur in residence, trying to help academics at a Cambridge Institute translate their research. After many years in the drug discovery, we've got the traditional drug discovery paradigm where it takes between 12 and 15 years to get a drug onto the market. Your patent life is 20 years, so at most you've got 5 to 7 years. The phases are split up quite simply across to early drug discovery, which is cheap in comparison to when you get to clinical trials, and that's when things get really expensive. It's probably about 3 billion euros now to develop a drug, and what you want to do is to stop research that's not going anywhere early before you start losing loads of money. The AI and the machine learning revolution, it's actually bringing something new to the table. You're able to screen huge data sets and mine things that you were not able to see before. And it's accelerating, the aim is to accelerate drug discovery and reduce the cycle, hopefully by 50% of more. And to be fair, most of the companies in AI and machine learning are focused on small compound discovery. Okay, that won't work. What you can see is that there's about 52 companies that are focused on chemical design, which is great. It was never a problem finding compounds. There's quite a few focused on clinical applications, and there's a plethora of other companies across that are facing some just very disease focused like immunology, some looking at target identification. Now, looking at the progress in AI and machine learning, I just picked out 4 companies that have grown bigger because they've bought out other parts of other companies. So they're almost end to end from target ID. And you can see that most of these companies are not even 10 years old, and yet they're in phase 1 trials, such as phase 1 for idiopathic pulmonary fibrosis. You've got Benevolent in phase 2 for atopic dermatitis, Recursion for phase 2 neurofibromatosis, and several others, and Accenture in phase 1. But the real question is, will AI designed drugs be more successful in the clinic? And I think We don't know the answer to that question. We know that Accenture did a collaboration with Sumitomo and Danippon Pharma, and they discontinued one of their phase 1's trials. Now, I went to the EBI before coming here at Cambridge at the Wellcome Genome Campus, and I talked to the people there. Where do you think the company's going and AI's going, and where the technology's going? And I also found this blog here where people were asked, do we think AI and machine learning companies have reached their peak? And the consensus was that 33% of the respondents thinks we're only at the beginning of the curve, and we're going to get there in maybe 9 years' time. So this is a really exciting space. But coming back to that question, why do things fail in clinical trials? And I took this from Fierce Biotech. It's probably a biased set of studies, but I think Fierce Biotech is a fun magazine to read. And they just picked up drug failures in 2020 and 2021. And if you look at the right-hand side, nearly all have failed in clinical efficacy. One failed because there's tox limiting dose. One failed because it was no better than the competition, which is fair enough. And if you look at it, around 60% are small molecule and about 40% are monoclonal biologicals. So what are the reasons for failure in the clinic? And taken from Sun et al., about 40 to 50% fail in clinical efficacy, 30% in safety tox, 10% PK, and 10% business decisions. And I just want to look at each of the 4 subsets as why they fail. And now I will be presenting my personal views on this, having been 30 years in the pharma industry and subsequently working on it in the future. Biology and the reasons for failure. I believe this is the fundamental reason we fail in the clinic. We don't always know what the right target is. Just because something is upregulated doesn't mean it's a driver of the disease. It could be that it is a consequence of the disease. Is there redundancy in the system? So you might choose a target to inhibit, but there might be loads of other variants and it makes absolutely no difference whatsoever because you haven't inhibited enough. The next question is, do I need an activator, inhibitor, or modulator? You might think that's a simple question, but it is not, especially when you get to receptors and you've got positive allosteric modulators and you've got things like biased agonists if you believe they exist. Is the disease a 1, 2, or 3 hit process? Let's go back to idiopathic pulmonary fibrosis. You've got patients and biological twins that have the same genetic mutation that predisposes people to full pulmonary fibrosis. One gets the disease, the other doesn't, so it's a 2-hit process. So if you're targeting the problem in the first hit, it may do absolutely no good later on trying to target what the actual mechanism is. You need human relevant disease data. It's no good trying it on mice. It's humans you're trying to cure. And there is actually a real opportunity for AI and ML and various technologies to determine the right biology. But underneath, I've said discovery scientists to beware. Kynurenine 3-monooxygenase is a target we worked on for severe acute pancreatitis. At the time we were doing this, AlphaFold 2 was not available. So we did it the old-fashioned way and set up a crystallization system. We found a weak inhibitor very quickly through rational design. In fact, it took the medicinal chemist a couple of days to come up with something. But because we had a crystallization system, we were able to show that compound bound in the structure, and it opened a teeny little pocket. So the chemists immediately designed more space there. And AlphaFold 2 at the moment will not be able to deliver those changes yet. But with time-resolved crystallography, we'll get there, but it's not available yet. And we're able to make a very low nanomolar inhibitor. A very unpopular part of science is enzymology and enzyme kinetics. It is a rare skill set. It used to take me years to find people specialized in this because they don't like it. It's really hard to get inhibitors, and here's your typical enzyme reaction, but it's not that simple. you've got your enzyme binding to your substrate, your enzyme substrate, your enzyme transition, your enzyme product complex. And unless you spend huge amounts of time trying to work out what the reaction pathways is, it's really difficult to know which part to target. We had a particularly nasty little cysteine protease, could never find any drugs against it. Again, alpha-fold 2 doesn't yet show that what the different structures are along the pathway. And it was the product, the release of product, that we found was the only way to inhibit it. And that was only because my boss used to love enzymology and differential calculus, and he'd spend hours and hours and hours deriving equations so we could develop the assays. And here is a particularly interesting example, and it's born from personal pain. It was not my project, but we lived through the pain with everybody else. It's an old example, but once you learn a painful example, you never forget. Pro-protein convertase subtlysin connexin 9, PCSK9, is the dream drug. Human genetics show that gain of function increases hypercholesterolemia, loss of function mutations, hypoglycemia. You can reduce it, you can produce it in mice. Fantastic drug. You could not have better drug validation. So we did a drug discovery initiated. We had some, it took a long time, but we got some excellent selective inhibitors eventually. Didn't work in vitro models. Didn't work in vivo models. What on earth is going on? We eventually mutated the catalytic site, and that didn't prevent anything. So clearly the MOA is not based on the protease activity, but on ligand interactions with the receptors. So the correct therapeutic is a protein-protein blocking interaction with the monoclonal antibody, and that's making billions of dollars at the moment. So the moral is, do not assume the obvious biology. Development reasons for failure, these should be rare. It's long been proven by many pharma companies that if you make something small and soluble with low lipophilicity and a dose less than 100 milligrams for a small compound, you're not likely to have much tox problems. You should be able to predict the tox. The thing you really need to do is fix the solubility. If something starts as brick dust, it'll only end up as brick dust. It is very hard to make something insoluble soluble, and I think there's a real opportunity here to factor in solubility predictions right at the beginning. Manufacture of small molecules is not a problem. People think about it and develop it well, but there are AI companies out there, machine learning, trying to accelerate the process. The real killer is biologicals and heterogeneity. They're produced in cells, and post-translational modifications vary. And we've lost, or I've been witness to 2 fantastic potential drugs that we've lost because we just could not fix the manufacturing. And this is because we didn't understand which post-translational modifications were important. You normally cut them off if you don't know what to do with them, but that isn't good biology. And we're actually just starting a new Max Planck group as a Dioscuri Center at the Mal Polska Center of Biotechnology, where for the first time somebody will be properly modeling what these post-translational modifications are. Clinical reasons for failure. Lack of clinical efficacy. I've already talked about you're doing the wrong target. You reach your dose limit. Well, you know what your dose limit is because you know that from tox, but you have to be really careful. And I would probably say don't bother putting it into the clinic because your patients can't tolerate it. IL-2, interleukin-2 therapy is really good, but it's got very severe tox problems and patients can't tolerate it, so they come off trials. So I would personally say don't even bother. We can't select the right patients, patient stratification. We need patient stratification biomarkers, and we need those early. And we also need biomarkers that study the progress of disease, because nobody is going to start a 5-year clinical trial. We need those biomarkers, and they in turn need their own clinical studies. And lastly is patient compliance. Patients are your customers. If you make a drug that they hate using, they won't use it. And patients, lovely as they are, they're very naughty as well. And it's been shown in many clinical trials they lie about what they do. So if you have a device developing a medicine, what the device say they do and what they say on their patient records they do not correlate. So you've got to make things easy and accessible and make patients want to use them. We're almost at the end. Business decisions leading to failure. This has been something I've been most sad about. There's always a change of strategy. Life cycle is about 12 years for a drug, and pharma companies change their business strategy every 5 to 10 years. So you never finish the life cycle before you change your strategy. And this is where I think maybe machine learning can have an unforeseen beneficial consequence in shortening the timeline to keep within the business cycles. Project prioritization, you've got too many projects, you can't fund them, but it's a great opportunity for biotech to pick up. Too far behind the competition, it happens, it's the right thing to stop. But something I want to alert you to, that the market is changing. Advanced therapeutic medicinal products are about to hit big time, and there's financial variability. You really need to know who will pay. Let's take the example of antibiotics. Everybody says we're heading for disaster with antibiotic therapy. It's not that we can't find good antibiotics. I've been on projects, we've had really good antibiotics, but nobody wanted to fund them because maybe you had a fantastic gram-positive, and if I've got necrotizing fasciitis, I don't care what you give me, I want myself cured. But it wasn't a pan gram-positive, gram-negative antibiotic. Arcogen bankruptcy is a real example of this. They found a fantastic new aminoglycoside. It hit all the resistant organism, and yet it went bankrupt. Why does it go bankrupt? Because you make an antibiotic and the doctors stick it on a shelf for emergency. Can't pay for it. Approved types of drugs, the paradigm's changing. I was at the European Society for Gene and Cell Therapy, and we all realize gene therapy is really good for monogenic diseases, but actively people in academia are hitting chronic diseases with gene therapy. And if I'm a patient and you just give me one injection and I'm cured, why would I want to take a compound once, 2 or 3 times a day? Watch this space and adapt. And if you look at it, a lot of the AI ML companies are focused on small compounds, but what's not focusing on is the biology, the phenotypic screening, the human assays. So conclusions. You understand the biology behind it, the disease is critical. Wrong biology will inevitably mean you fail in the clinic. Closer collaboration with academic community and industry is critical, because you can have thousands of researchers all around the world working on deep biology. You're not going to get that in companies. ML and AI can clearly accelerate the drug discovery, but it needs to adapt. And to my mind, I think it really needs to focus on the deep biology. And one key learning is all these companies employ some really skeptical people that ask stupid questions, the simple questions and the difficult questions. So thank you.
Thank you very much. It was really interesting. And now moving into lectures that will be given in a topic of phenotypic drug discovery, but from a different perspective. So if you think about phenotypic drug discovery and you look at the healthy versus diseased phenotype, and you look what is changing between those phenotypes, those can be small molecules, biologics, so large molecules, or even you can go a level up. So look at the microorganisms. So now we will dive into the fantastic world of microbiome. And first of all, we'll have Dr. Paweł Łabaj talking about a fantastic project Metasap. Dr. Paweł is from Małopolska Center of Biotechnology from Jagiellonian University. And after Paweł will go into a deep sea of microbiome of the skin with Dr. Chris Calvert from Ghent University. Chris is a specialist in skin microbiome and metabolome. So, Paweł.
Thank you, Kaya, for a nice introduction. Okay, so Danuta was talking how it's going in the drug companies, but in fact, first we have to have a concept, and it was mentioned that it's good to dig into the big data, right? So, but we have to have somewhere this big data. So what I'm going to talk is where we can look for our possible compounds, because of course we can make many reactions, but the best bioreactor is just the mother nature. So, and other thing which is also very important, that what I'm looking at is just going to the research from this data-driven perspective, going to the data science, because then once we know this understanding of the disease, of the processes which are going on, then we can switch to the hypothesis-driven. And for a long time, it was that you always need to start from the hypothesis-driven, which is now also required by the funding agencies. But unfortunately, in many cases, the data-driven, it's a way more perspective. So maybe quickly how it has started. It was like about 10 years ago, then a friend of mine, Professor Mason from New York, was riding with the metro with his daughter. I think she was 3 or 4 years old then, and she was eating ice creams. And then she has asked, "Can I lick a pole?" Like in the metro in New York. And he, as a father, wanted to scream, "Of course not." But as a scientist, he just started wondering, why not? I mean, what's there that we cannot get in? So he started then, after going back home, he wanted to read about this, and he has figured out that all pathogens which we know, everything what we know bad about bacteria, is only those we grow in a laboratory in a huge amounts, and then we tested it. We have completely no idea what is outside. So he started sending students in the New York metro system to collect some swabs. And after a few years, he just put everything together and they just published a paper. And I think the most interesting discovery was that for the half of the sequences from the sequencing, we have completely no idea from which organ they're coming from, because you cannot cultivate them in the lab, so we have completely no idea. They've seen also many other things, like there's a difference in diversity when you compare different parts of the New York. But for him, it was like the trigger that maybe we should do something this direction, and maybe we should go further. And as Chris is saying, of course you find plenty of pathogens, but they're on the small amount. So he say on one of the TED-Med that I would advise any new parent to roll their child on the floor of the New York subway. It's like the super multi-vaccination at just one shot. What they found is that, of course, it's very close to skin, what they found in the subway, because people are riding the metro, that's why what's there. But in principle, as other professors are also saying, dirt is good. So, but whether maybe this is just a New York dirt which is good. So this is what Chris was start thinking about, and because I keep working with him with the other consortium, so he start asking people he's collaborating with, do you want to join? I mean, we know New York, we want to see how it's in the other cities. And I say, why not? And this is how the MetaSub was born. So I joined when I was in Vienna. Now we are doing this in Krakow. So as you see on the map, at the moment we're covering the whole world, almost every bigger country, all the small countries. So at the moment we have over 100 cities all over the world and over 1,000 participants. And every 21st of June we are collecting the swabs. Also in Krakow we are swabbing in 3 tram lines, because unfortunately in Krakow we still do not have a metro. But we are getting there. So this is what we are collecting. This is the huge amount of data which we are slowly getting. And what are our aims? I mean, those are our aims, that we want to standardize collection, processing, and analyze, because otherwise, what's the point of having the data if we cannot compare data from one place to the other? So this is, let's say it's done. We are sort of done with the critical spatial metagenomic and forensic genetic maps. I will say a few words there. But I think what is the most important in the context of what we are talking here about is the identification and tracking antimicrobial resistant markers. And in fact, we are now running quite interesting challenge about this, where we, knowing the resistant isolates from the hospital, from the one city in US, we want to see whether we can find the markers of exactly the same resistance strains in the whole US, in the different cities, for the few years. And also what is slowly happening, but this is still, we are not there yet, is identify novel biosynthetic gene cluster for drug discovery. Because the nature, it's doing way more reactions, experiments in the 1 second that we can even imagine. Okay, so having those all data, of course, you can focus and you can start thinking, but at some point you will be very biased and your whole team will be biased with your original thinking. There was a study showing that the best papers are when the old professor is dying, because all the others from his team finally they have a freedom of thinking. So going this direction, we're taking all those data and within the critical assessment of massive data analysis conference, we are just giving to the society, find something interesting in those data. We are not giving you, it's not a challenge when there is a ultimate goal, just find something interesting. And here we just gave the data from the perspective, from the forensic perspective, whether we can identify from where the sample is coming from. And over 4 years, there were over 20 manuscripts. What was the outcome is that it doesn't matter whether we're looking at the data from the taxonomic point of view, functional, or just pure sequence. I mean, the k-MER-based composition, it's very easy to say from where the sample is coming from. But what is interesting is, but we cannot predict if we don't have the specific location yet in the database. And it's coming from the fact that the microbiome is connected with the ecological niche. It means it's not like the GPS that if I know this, the 3 positions that in the middle, okay, I know where I am. Here, if I know composition, that's 3 different locations, I have completely no idea what is in the middle, right? If it will be higher level, lower, close to the lake, it can completely change it. So we cannot do this. So the CAMDA was 2017, 18, 19, 20, and then it was came time last year when we finally put together all the data and managed to publish the results. It appeared in Cell, over 5,000 samples. So we identified that there are core taxa which are everywhere. We find that there are specific microbes, but we also find, let's say, almost a 1 million CRISP arrays, which can be later investigated for the different diagnostic procedures. We also found a huge amount of the new viruses and some new bacteria. Oh, sorry. But the highlight is that we see that antimicrobial resistant genes are widespread in the city. I mean, we see them everywhere, just on the very low level, somewhere on the very low, somewhere on very high. Together with other papers, we can say that, okay, it depends how strict are the regulators in the specific part of the world. But we see it's everywhere. It doesn't matter whether we're looking at the pathways or whether we're looking at the cities, we just see that the resistance is everywhere. And also when we look into what is in the air, because previous was from the samples from the surface, we also see there, and interestingly, in air you see more than on the surface. So what else? We also look into the viruses and we see that they are very specific. They are even more specific than the bacteria, but still you can combine different cities around the world, probably where you have a lot of the human transmission transfer. So probably if you will correlate it with something like the logging to the Facebook profiles, I guess it will be very highly correlated. So this is where we more or less where we are. So what could be the next? We now know that you can investigate many things by detecting what is there in the transit system, especially when you have like the hub, transportation hub, the big train stations or the bus or the metro. So it should be, and in fact the technology is already there. I mean the pieces of technology that you can think about the global pathogen early warning system, which could alarm you early enough that the city and the decision makers, they can think about and prepare that the new superbug is in the city. Now we know when somebody is in the hospital and probably he already spread this bacteria once he was for 3-4 weeks walking around the city taking the antibiotics which were not working. But if we could imagine that we have a device in every bigger transportation hub, which is just constantly monitoring what is in the air, the decision makers will have a few weeks of advance to prepare to what can come. And just to summarize, a few years, 20 years ago, it was said by the Sidney Brennan that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So the ideas are at the end. First, we have now, it was shown on the previous session by Jack, that there we have a now very powerful sequencing technology. So we have this new technique. It allows for the new discoveries like those which we have now in the MetaSub. And thanks to data science and the exploratory of those data, we can have now the new ideas, what we can do and how we can, let's say, apply it in the drug discovery. So thank you. This is our dream team in MCB. So thank you for your attention. And just last week it was, if you want to listen or read a bit about the history of MetaSub, we just got a paper in iScience about how everything has started. Thank you. So we have now time for questions, or will it be during the panel session?
Now we ask for Chris Cavallard session, please. It will be online. And
Hello. Hello, my name is Dr. Chris Cavallard from Ghent University. I study the skin microbiome in health and disease. Apologies for not being there with you at the conference, but I'll try to bring my talk as best as I can here online. So I will talk about the skin microbiome from lab to treatment. So how to use the power of the skin microbiome, not just in the lab, but really to use it as a treatment for existing disorders. First, I want to talk about atopic dermatitis, eczema. It's also a very good example of where there's a very clear impact of the skin microbiome in general. First, a bit of background. It's a chronic relapsing inflammatory skin disorder. It results in these red rashes on your skin. It can happen at any age, but typically occurs in childhood. The environment plays a role. For instance, contact with certain chemicals can play a role there. The skin microbiome is very important. If it is broken, for instance, with a scratch, it's difficult to repair itself. Also, the skin immune system is very important here. Immunosuppressive medication can also suppress the problem. And last but not least, the skin microbiome plays an important role. We see that there's a lot of Staphylococcus aureus on atopic skin. Such antibiotics can help, but it's not really advised, can lead to resistance. But it's skin microbiome plays a crucial role here in this inflammatory disorder. This is the pathophysiology of atopic dermatitis. We have healthy skin and we have a healthy skin microbiome. The skin microbiome is provoking an adaptive, a balanced adaptive immune response, and it also maintains the healthy barrier there. At some point, there's a barrier defect. It can be a scratch, and that scratch can lead to a dysbiosis in the skin microbiome. So things can go heavily off. And that in itself is causing an inflammation. Typically, that's a Th2 immune skewing. And with that, we also see that the skin cells are no longer able to produce antimicrobial peptides, which is again not ideal. And that is causing the pathogen Staph aureus to increase on your skin. And we all know that this one is definitely not a good one to have on the skin. It will maintain the skin condition and it will not resolve the condition. Now, we've tested the impact of the skin microbiome when you would inject the wonder drug dupilumab in the skin. So this is really the wonder drug we can, for moderate to severe eczema. it really has no antibacterial properties. It's injected in the skin, but when that is done on week 4, week 8, week 12, week 16, you can see that the red part is really decreasing over time. The red ones are Staph aureus. So it's really, it's reduced by half by simply injecting a molecule which suppresses the immune system. So you suppress the immune system, and as such, the microbiome is really also impacted there. So if you aim towards a stable immune system, also the microbiome stabilizes itself as well. And that results, of course, in less lesions, and the skin condition disappears. So that's a beautiful example that there's really a crosstalk going on between the immune system on one hand, within our body, and the skin microbiome. The immune system can influence the microbiome, but also the other way around. The microbiome really impacts our immune system on a daily basis. We can, on one hand, resolve the immune system by injecting this drug, and as such, the problem is resolved. Or, on the other hand, we can also modify the skin microbiome, where then again this will have an impact on the immune system. And there's also a startup doing this. Startup is really applying good microbes on the skin with eczema, so that eczema would be resolved. They outcompete the bad bacteria. They would outcompete Staph aureus, and as such, they will come back to a new balance, to a better balance, with a much better immune system, much better microbiome, and such it can resolve the skin condition. So that's crosstalk between the immune system and skin microbiome is really important there. Now off to another skin condition, if you can call it a skin condition, underarm body odor. I'm really well known for this. I've been researching this topic for over a decade, so I know a thing or two about body odor. And this is actually a sample of more than 200 people. Everybody's armpit is somewhere here on this scale. If your armpit is more on the left, you have more corynebacteria, more of the red ones. If your armpit is more on the right, you have more of the staphylococci. You also have less body odor. So having body odor or having no body is really not a black and white story. It's really more of a gradient that we see there. Everybody's armpit is somewhere on the scale, and it can move a bit from time to time, but in general it's quite stable. But at some point people suffer from body odor, and they really find themselves more on the left side, and it's really hard to get back on the right side again, because the skin microbiome can be very stable over there. So our idea was to change the skin microbiome, to steer towards the right by putting new bacteria there, other people's bacteria there, in order to resolve the situation and in order to resolve body odor. And we've done that by a theory called an armpit bacterial transplantation. We are putting a healthy and non-odorous underarm microbiome on the washed armpits of someone suffering from it. And that usually comes from a relative, the mom, the dad, the brother or sister. This is the very first successful armpit transplantation. This is actually a DGGE, it's old school molecular technique that visualizes the bacterial abundances and species. Every band is basically a bacterium. The more darker it is, the more abundant it is, the more they are present. A bit of a background, so this is actually an identical twin. You could hardly distinguish them based on sight, but based on smell, you can very much distinguish them between the two. So I found it a really interesting case. I approached them and said, yeah, can we swab your armpits? We did, and we saw that, okay, one twin had a lot of the corynebacteria, a lot of the smelly bacteria. The other person had lots of the staphylococci, lots of the good bacteria. Said this is an excellent case. Can we exchange bacteria here? So from the non-odorous armpit to the odorous armpits. They agreed, and so we did 3 transplants on 3 consecutive moments with a couple of days in between. We washed it and then we applied as fresh as possible the other person's bacteria. And we could see that the good bacteria were able to be introduced. So they were able to, we could put them there, and they were able to stay there over time. Not just that, we were also, and that's more importantly, we were also able to decrease the diversity. So on the right-hand side, we would decrease lots of the diversity, lots of different corynebacteria were able to be suppressed or removed. And more importantly, the abundancy, the darkness of the bands also decreased a lot over time. And that resulted in a much better smelling armpit than before. Even after a year, the situation was still under control. He still had staphylococci, the good staphylococci there, and the corynebacteria were still under control. So a perfect example that you can change some condition with the microbiome. Now, the armpit bacterial transplantation works, but it's also a lot of work to do. They all need to be physically present. We need to make sure that the donor is correct. We need to screen, etc., etc. So we moved up to the next phase. We were able to put the good species into a spray can, into that we call this probiotic spray, so that they could do their own transplants on their own means on a daily basis or every 2 days if they want. So that's much more handy for everybody as well. So we did a clinical trial on this spray on about 79 people. And these were the results. Initially, this person had a lot of the corynebacteria, very smelly armpit. So he had like a -6 on the scale from -8, which is very unpleasant. 0 is neutral, +8 would be perfume. So -6, yeah, you would really smell that person if he crosses your paths or if you're in the same office with him. It's a very bad odor, and he was suffering from this for almost his entire life, adult life. Nothing that he tried or sprayed on would really help. So that person started using the good bacteria, started applying the good bacteria. We could see on the DGE that the bacteria were able to be introduced. And that also led on the right-hand side to a decrease of the corynebacteria, both in abundancy and diversity. And that also led to a significant improvement in hedonic value. So it went from a -6 to about a -2. And also afterwards, when that person used a placebo with nothing in there, the good bacteria were able to still stay there. And we could see that a lot of the malodor-associated bacteria were actually removed from his underarm. And that also led to big improvements in underarm odor. So initially he had a -6, and now on average it has a -1. It's not completely gone, but it's so much better than before. That person was also super happy. He says, my wife is happy, my clothes smell better, my pajamas smell better, my jacket smells better. His colleagues were super happy. I mean, yeah, super happy. They also noticed it. So that was a huge improvement for him personally as well. So this is, with this short talk, I want to show you the promise of the microbiome, the skin microbiome, that you can really solve conditions by changing and improving the skin microbiome. We wrote a big review on this topic that sort of summarizes all the skin microbiome transplantation and manipulations. There's a company in Japan, Biogenomics, that is applying good staphylococci on the skin to improve appearances. We're doing armpit microbiome transplants against body odor. There's a company in San Diego, Matrices Bio, that is applying good bacteria against eczema. So I talked about that earlier. So they're really applying good species on the skin to resolve eczema, atopic dermatitis. There's another company as Biomed that is applying good Cutibacteria on the skin to resolve acne. There's also other bacteria, other companies that are applying non-commensal bacteria on skin to improve the skin. There's also a company in America that is applying genetically modified skin bacteria and using it as topical therapeutics. And there's also a series of other companies that are using lactobacilli or bacilli on the skin for to improve certain conditions. both dead and alive. So this has all come up in the last 5, maximum 10 years. It's a very recent thing, but has lots of promise. Skin microbiome is really crucial in training our immune system, and it's also crucial in the manifestation of skin conditions. So by creating a stable skin microbiome, you can also improve skin conditions. These are my contact details. I thank you for your attention, and of course you can reach out to any questions, but I will also definitely be there in the questioning afterwards. Thank you.
So thank you very much, Chris. It was a fantastic example of phenotypic drug discovery. So you had a phenotype, a smelly one, and then the not smelly one phenotype. A fantastic example. So now we'll move to more of small molecules and images. First of all, I will start with the introduction of the company, and then I will give a floor to our lead molecular biologist, Magdalena Otrocka. Starting with the company. So we are representing Ardigen. It's a Polish company. Well, I need to now to, okay, check if that works. Great. So we are a Polish company founded in Poland, in Krakow. We started operating in 2015. So lately we moved from kindergarten to primary school. So we finished 7 years old. So Ardigen is now 7 years old. We started with like around 15 people. Now we have over 170 people, and we are science-based company, so we have over 60 people with titles, so PhDs. We work mainly with United States, so over 52% of our revenues come from the United States, but we work also with Europe, with Australia, New Zealand, with Japan, so all over the world. We already collaborate with 9 big pharmas. So our main mission is to improve health. And to improve health, we're working with biotechs, big pharmas, to increase, to boost with AI the drug discovery process. So in general, we are covering all the drug discovery process, and our main operation is AI services. So we work with AI bioinformatics, but we have a specialist in chemoinformatics, biologists like great biologists, immunologists, software engineers, data scientists, and we work mainly as AI CRO, but we have also specialized services. So microbiome mentioned here already, but also immunology and biomedical imaging, the topic that will be covered in a moment. What is more, we also received grants that are supporting a phenotypic drug discovery. So the microbiome biomarker discovery platform mentioned as the first one. Here we have worked with the patients, with data from oncological patients, that we managed to build a module that can find differences between diseased phenotype and healthy phenotype, and it's based not on the taxonomy, but it's based on the genomic factors that are behind that. And it's a deep learning method that uses vectors to describe the microbiome that we found in the gut. Bioforte technology is the one that we are now finishing the grant, and we've built also the technology here more to look for probiotics, like the strains that can be used then to change the phenotype that is not responding to a therapy to the one that is responding to a therapy. And the last but not least, High Sky grant, it's the development of a cell-based phenotypic platform. It's based on high content screening, so imaging systems that uses artificial intelligence data in artificial intelligence algorithm to boost also the drug discovery. Thanks for European funds, we can also develop technologies that drive, boost, leverage the drug discovery based on phenotypic screening. So please welcome now Dr. Magda Otrocka. She will bring you closer to high content screening technology.
Thank you, Kaya. So as Kaya said, I will now focus more on phenotypic screening and imaging. And I would like to start what was already expressed today by Danuta this morning, or in the first presentation. And once again, talk about the fact of how the drug design, drug discovery process looks like at the moment. Everybody knows it's long, expensive, and inefficient. First of all, when we start our discovery project, we have a limited chemical space that we start with. Even if the big pharmaceutical companies have libraries, chemical libraries of several millions compounds. Doesn't work now. Yeah. Sorry. So this is just a small portion what nature can offer, and also what we can theoretically synthesize as chemists. And then when we use those libraries to screen for the biological activity, we obtain between 0.1 and 5% of hits. But what is even more important, those hits, so biologically active compounds, are usually of poor quality, and many of them never reach a clinical phase. And even if they do, then the success of the clinical trials, it's a very low percentage. So there is a lot of debate and also efforts to improve this drug discovery project, drug development pipeline. And there are many approaches, and the one that we believe in RTGen is integrating artificial intelligence in drug discovery projects. So our approach to do it is to use images to discover new better drug candidates. So looking and trying to find molecules that are transition the disease phenotype of the cells to the healthy cells. And we already, so our study already showed then this approach can significantly reduce the cost, time, and also give a better candidates. So for example, we can start with a virtual means. When in silico, we can test or select the molecules from the much bigger chemical space, like up to 10 million compounds. And out of selected compounds, so this virtual screening, then when we test them in a wet lab, we can improve the hit rate up to 40%. What's more, we can also use the technology during the optimization of the obtained hits. And we already have cases that we showed that what normally in the lab is done in the several interactions, we can reduce to the 2 cycles of the laboratory. What's more, this technology can identify not only desired phenotypes, but also the phenotypes that are show us that the perturbation, the small molecules, has a side effect on a very early stage of the process. And we use this technology to to increase predictive power of cell painting assay. And I will now explain you, or in a minute, we'll explain you what the cell painting assay is. But before I do that, I will give you a brief introduction to the technology that cell painting is using. The technology is called high content screening, and it allows multiparametric detection on a single cell level. This technology is around 2 decades now on the used for 2 decades, and it simply combines the automated fluorescent imaging, automated fluorescent microscopy with the quantitative imaging analysis of images. So in a typical with a typical high content screening project, we start with the same step as any fluorescent microscopy experiments. So we need the cells going in vitro, stained with fluorescent dyes or fluorescent proteins. And then we take the images of those cells with the special microscopes that are allowing to very fast acquire terabytes of the data from the mito-treated pellets. And then we need to analyze images. In a classical approach, those images are quantified using specified software that can translate the images, what we see by eye, to the numbers. And we can obtain from this, as an output from this analysis, we can obtain several parameters that correspond to a single cell, the fluorescence of the single cell, but also the morphology of cell, and also subcellular localization of our signal. So this is the, let's say, classical approach. And being AI company, I cannot mention that now there is a growing interest of applying deep learning and computer vision to analyze images. So it's not yet fully established, but it's very much used. But independently on which analysis of image analysis you are using, what you get from the high content screening is this single cell readout that can measure subcellular events, but also look at the phenotypic changes. And here in this slide, I summarized and wanted to show you a couple of examples of high content screening assays, high content screening detection methods. The very first one, transfluor assay, that was developed, the first high content screening assay developed to study GPCR receptors. Any other classical example of high content screening assay, it's looking at the morphology of neurons, neuronal cells, neurite outgrowth, or such events like autophagy and many others. So what is common with this assay is that we can now, in a high throughput screening, in a high throughput mode, assess the targets that were not able to be studied in such a throughput before. But what's even more with high content screening, we can use it also for screening, phenotypic screening of the target agnostic screening. And here it's where cell painting assay comes to the stage. This assay was developed and published in Nature Protocol in 2016. It was developed in Ann Carpenter lab from Broad Institute. And it uses quite simple simple concept of staining several cellular compartments, the key player in the cell machinery organelles like nucleus, endoplasmic reticulum, nucleoli, cytoskeleton, and mitochondria. And then analyze those images and extract all possible features or parameters that for each of the stain. And this, the assumption is that those features will differ if the phenotype is different. So can distinguish, those numbers can distinguish between different phenotypes. So if you treat your cells with small molecules or with the genetical perturbations, and you do it in a high scale, so have several mito-treated plates with those cells, then you image them with a special microscopy and analyze images and extract all these features. So it can be several hundred of the features, and then put them together, cluster them together. So all the phenotypes, so what you see in the images, similar images, should cluster together according to those features. And you can use this approach to predict the bioactivity of new compounds, test or look for the mode of action of compounds it can help you to prioritize your hits, characterize new synthesized compounds, look for new phenotypes, or even redefine your screening collection. However, to be able to do all this, you need a set of reference images with the perturbations that you know what the mode of action is. And several big pharma companies and also small laboratories started to produce such data sets, but it's not affordable for everybody, especially for academia or small biotechs. So this is why this JUMP-CP consortium, so Joint Undertaking in Morphological Profiling Consortium, was funded in, I think, 2018 or 19. And again, the merit of Ann Carpenter, who convinced 2 big pharma companies and 2 non-profit organizations to build such a public data set to scale up image-based drug discovery strategy. So JUMP Consortium works also with some collaborators and 6 supporting partners, and I'm really happy and very proud to say that RDGen is one of the supporting partners. We bring also our technology to build this resource. And this is also, they will be publicly available soon in the 1st of November, and will contain, so there will be images corresponding to 120,000 compounds and also the subset of genetic perturbations, so around 200 terabytes of the data. And this dataset can be used then publicly for determining the mechanism of action, prediction of compound activity, toxicity, and also matching the drugs to the disease state. And this, we believe, or all people involved in the JUMP consortium, believe that will relieve a major bottleneck in the pharma pipeline. So finding the compounds that are efficient, so they have certain efficacy that can be then demonstrated in a clinical state, as Danuta mentioned also during the presentation, her presentation, that is really a problem for the drug discovery. If you want to know more about JUMP consortium, please look at the website. But now I will explain you shortly how we use our technology, so how RDGen helps GP Consortium and uses cell painting for leveraging the phenotypic drug discovery. So our approach is to use the computer vision and phenotypic analysis of the images, and then combine it with another AI approach for analysis of the chemical structures. We combine them together, which gives a better prediction power, a better analysis results. We can also apply on top of that multi-omics data and bioinformatics data that we hope will be even better. But here I have in this slide example of how this combining of the images and structure representations can give you a better results in your prediction and improve the prediction power. So here, those pictures, those UPM maps show you the analysis, the results of analysis of the public data set, in which we found 2,000 compounds of known mode of action that we assigned to all those compounds. And here in these pictures, there's few selected to show you, and they're color coded. And the very first from your side, it's the left picture, is showing you the clustering of those or representation of those images and compounds based on the human-defined features, so the analysis, image analysis quantification. As you see, we get kind of one big cluster and 2 smaller clusters, and then we have all the colors everywhere. Then if we cluster the same data according to chemical structures, we have a bit more clusters, but still colors everywhere. However, we combine when we use this our approach. So the deep learning analysis of images and the chemical structures, then we have much better clusters, and also the colors are according to the structures. So it looks much, much better. Another example, it's the platform that we have built for one of our customers, big pharma company that actually tested the big part of the chemical library, around 1 million compounds, with the cell painting assay with the multiple cell lines and at multiple concentrations, producing 200 terabytes of data. And they have also another data, biological data, but not for all the compounds, for the part of the compounds. And what we try to do is to predict the activity of compounds that were not tested in a certain bioactivity assay based on the cell painting data. We try to predict if they will be active or not active. And this is quite a big success. The customer is very happy with the results. And this can really reduce the number of wet lab work, because they don't need to test all those compounds in expensive assays, but those selected that are predicted to be active based on the cell painting assay. So the conclusion here is that integration of artificial intelligence in a phenotypic drug discovery process has a very, very big potential to reduce cost, time, and efforts needed to get the good drug candidates on a very early stage of drug discovery. And our artificial intelligence models can be used at any stage of the drug discovery process, starting from designing library through the virtual screening, then identification of target and on-target effects, and assigning mode of action to hits that were identified during phenotypic screening, and also optimize compounds to obtain desired phenotype. And at the very end, I wanted to thank all the people from our biomedical imaging unit, Michał Warchoł, who is the head, and also the group of people working with computer vision and AI computer vision. We have also a chemoinformatician and software engineer. So thank you.
So now we'll start the panel, and Paweł will lead the panel, but we have also our online panelists, two of them. Professor Janusz Bujnicki, who is a specialist in bioinformatics, really. He's a professor of biological sciences and head of the Laboratory of Bioinformatics and Protein Engineering at the International Institute of Molecular and Cell Biology in Warsaw. And also Chris will join us for the discussion. So we will wait them to join. Oh, yes, fantastic.
Yeah, we have them.
Yeah, Paweł, please.
Yeah, so in addition, in our discussion, we also have Magda Winiarska, CEO from the Biomed, Polish Association of Innovative Medical Biotechnology Companies. And in fact, this discussion which we are going to have now is about how to make phenotypic drug discovery more efficient through academia-industry collaboration. We've heard interesting talks. I mean, mostly the focus of the topic was the last talk and the first talk. The two in between were coming from showing some ideas from different perspectives. What is the most important in this topic is to identify that during the drug discovery, we have many steps. Some of them can be shortened thanks to the AI, but some cannot. And I think it's very clear to specify that at the moment we are not able, I mean, with the current development of AI, how we're thinking about AI, I mean, many cases AI is as a buzzword, right? We say AI, but it in many cases means, in fact, that we have just the old type data exploration using the different machine learning approaches. And in this case, we can automate many steps, we can make them faster, and thanks to this, we can shorten some of the bits, but still some cannot be speed up. And maybe I would like to ask one more question, and just open the discussion, because it was explicitly shown by Danuta where we can speed up, how we can, these things we can sort out. And what about the toxicity? I mean, if we have set of the compounds and we find the best compound, but from the screening we see that it's toxic, and I think we can use AI to think about whether we can find a homologue which will be less toxic or even not toxic if the time of the application will be short enough. So I don't know, Danuta, do you want to start?
So on target toxicity, when you develop a drug, you often have many compounds that are ready to go into development. And if you see that one is showing some toxicity, you have to ask your question, is it on target toxicity or is it toxicity due to the compound? So you repeat the studies with other compounds, and if you've got many different structures and they're all giving the same effect, you're pretty sure you've got on target toxicity. So the ideal decision is you stop, you go back and you find a different target in the pathway. Sometimes you can't predict toxicity, and we study, for small compounds, you'll study a rodent, and you'll study increasingly pig rather than dog. And pig is closer to human than dog in many ways, but you still get caught out. And the other thing is, we don't always understand the biology of the disease. And you've heard me say this many times before, the time of dosing, not all drugs are expressed at exactly the same time. 15% of the genome is done expressed in a chronobiological order. So if you've got off-target toxicity and you know that your target is not expressed at the same time as your toxic, you've actually got a much bigger therapeutic window. So what we do is we plow ahead into the mechanism, but we don't explore the whole biology.
Okay, thank you. And Kaya, I think you also have some things in this direction.
Actually, I have more of a question here, because like more general one. I'm from the company side, and we have here two people that I would like to ask how it is from both from university point of view and here from Polish Association of Innovative Medical Biotechnology Companies. Do you see, Professor Janusz Wójnicki and Marta, can you see the collaboration between the university from both sides and companies and industry side in a way to make that drug discovery process and the phenotypic drug discovery and things that Professor Danuta mentioned more efficient through the collaboration here?
Yes, I think I was first. Good afternoon, and thank you for inviting me to this panel. Well, of course, collaboration between industry and academia is possible because it is happening. So it has been proven that it's possible. One consideration that needs to be taken into account is not only which kind of questions can be addressed in the industry or in the academia or both, but also which kind of questions are interesting for one side or the other. Some of the types of development, some of the types of questions are perfectly addressable by academia, but they may not necessarily be interesting for academic researchers, especially if they involve answering the questions that do not lead to some new technology or do not lead to immediate deep understanding of the process, but rather concern only just the selection of some entity at some level of the development pipeline. So this actually requires the very different schemes of collaboration, whether this is a scientific collaboration or a service. And as soon as all the processes in the pipeline actually cover the interests of both collaborating parties that have the relevant expertise, then obviously this can be done. And here For instance, the partners from academia, I envisage that they would be mostly interested in developing new methods, new predictive methods, especially if they can be used not only just for one specific project, but if they could be generalized to many different problems of the same kind, as well as solving deeper scientific questions. My background is in biology, so partners collaborating from the side of the biology would be probably more interesting in collaborating on solving problems where the academic side would contribute to solving some deeper problem than just leading to a drug, but providing some deeper understanding into the biology relevant to the whole process, where the drug, from the academic point of view, is just one of the effectors and not the most important thing in the world.
It's very great that I can take a part in this interesting discussion. First of all, I need to tell you that I'm not the scientist, so I'm not pretty sure if I understand well enough what is phenotypic drug discovery, but I really well understand what is collaboration. And the question is not if the collaboration is needed, but rather how it should look like. Did you know that the number of projects focused on drug discovery and drug development is much lower than some years ago? And the question is why. This is first of all what was said today, because it is really it costs. It costs lots of money. And the second thing is that what was relatively easy to discover, easy to develop, it was already developed. So today we need the effect of synergy to develop new drugs for patients. So the question is how, in this context, different entities, different parties should cooperate. It's not only about the cooperation between academia side and business side, but also between different types of businesses like RDGen, which has AI technologies and machine learning technologies, and other companies which deliver different types, different elements of the drug discovery process. But I'm pretty sure that cooperation between academia, business, it's today not enough to ensure that we will have new drugs for patients. I think that COVID pandemic showed us pretty well what is needed to make the entire process of discovery, of development faster and more efficient. First of all, it's collaboration between academic and business, but second of all, it's also engagement of regulators and engagement of the government, meaning transferring some financial sources to this type of projects, because it's so expensive, it's so risky, and we still have so many unmet health needs that we absolutely need engagement of all parties, because cooperation between business and academy, it's needed, but it's not enough to make this effort efficient.
So in going this direction, I mean that we need to have a support and collaboration. I think it was shown that we are starting, like say, from thousands of compounds. Thanks to AI, maybe we can start with even more and then quicker get something. But at the same time, it might be that thanks to collaboration and joint projects, we can investigate to better understand the biology of the disease and what we need. And then we don't need to start from thousands, but maybe hundreds. But it means that there need to be a way that the companies who are interested in the specific area, they can attract academic partners with external funding. That, hey guys, we are thinking about this one, but we have a good understanding which direction we should go. But maybe if we will dig deeper into the basics of the specific disease, we can find that half of the drugs which we are at the moment, at the beginning, thinking about, it just completely doesn't make sense. Yeah, and we don't need AI here. It's just like, no, understanding the basic stuff, and then if we can do like something like this pre-project, which will just limit it, then we, thanks to AI later, we can also shorten all the other tasks even faster.
Chris, what do you think? Because you've shown during your presentation the work between the university and companies. So what are your thoughts about that?
There's a lot of nice research going on in universities. I'll quickly do like this. And the beautiful thing is, well, every time I speak with industry, they say, okay, there's a lot of nice research going on in academia, but nothing seems to be happening with it. Academia is going a bit slower than in industry, that's for sure. And it's a fact that academia is doing very nice things. We're writing good patents, solid basis are being formed. And usually the idea is that if you have enough patents, you can create a spin-off with it. So that's what we're also doing here in Belgium and Ghent University. We have a couple of patents, and then we move towards a spin-off, and then we make a viable commercial entity out of that. It can also go the other way around, that a company reaches out to the university and agrees on a license term. So they would pay royalties to use that patent. But it's then again, the deal has to be made with the university and involved researchers. Academia is definitely open for collaboration with industry. Many academics end up in most of them end up in industry anyway. So in that sense, there's definitely a good crosstalk going on. Although there's also room for improvements. As what I heard is that academia is going a bit slower, and then they complain about that.
Okay, thank you. And from the Polish perspective, Janusz, how do you see it with the spin-offs and things that Chris just mentioned?
Well, yes, of course, the spin-offs are an important way of commercializing the discoveries done in the academia, the universities, and in the research institutes, but not the only one. What is important is actually the entire ecosystem. And in Poland, this ecosystem is not as strong as in some of the other countries, where there is a multitude of different companies from very early stage companies to more advanced spin-offs and spin-outs to middle-sized companies to big pharma, and people are actually circulating between the companies and the academic institutions. And in addition to people doing research, there are also other experts from the side of business and all the other expertise that is required to turn a scientific discovery into innovation that goes to the market and is successful there. So this entire ecosystem is required. And in Poland, we've been trying to build it slowly and slowly. It has been growing, but from what I heard recently, it has not been as good as before. So that then the number of spin-off companies is now, I mean, the growth is slowed down by many external, because of many external factors. But this is absolutely essential for the innovations to succeed. So it is not, so having a scientific discovery and just starting a spin-off is not possible for a success if it's done in a sort of business desert. So there needs to be an entire ecosystem for any particular discovery to be turned into innovation, unless of course this innovation is licensed or sold to some external partner abroad. And also, in addition to the ecosystem, what is absolutely essential is the experience, and not so many scientists in Poland, especially in academia, have experience in dealing with the industry. So there is no, and without the experience of partnership with the industry, there is no trust, and there is no understanding of what are the needs and differences with the industry. Of course, industry is and must be faster than academia in doing specific focus work, But in academia, most of the research is not really as focused as in the industry. It's not focused on the business goal. In academia, usually the research is slower, but it's so much broader. And for the academics to be good partners for the industry, it requires understanding, usually only that can only be gathered from experience, that in the collaboration with the industry, one has to get focused on a specific task rather than to keep going as broadly as is a typical attitude in the academia. And also the industry partners must understand, but they usually do understand, why the academic researchers are usually going broadly, because it's because of the breadth we can have unexpected discoveries that are serendipitous and not coming directly from the very focused question. So mutual understanding requires experience and openness, but this of course is dependent on the number of potential partners. So the more partners are available and open for collaboration, the better. And I would like to encourage everyone to try. And I mean, every academic researcher who's doing anything related to the industry and industrial application or commercialization of research, to at least try collaborating with the industry and then develop his or her own opinion on how it's going. Is it good for that person or not?
So I really want to echo what Janusz said, but so I come from the UK and I came to Poland in 2015, and part of that was looking for collaborative projects with GSK. And I sat somewhere where I won't name, and I sat in front of a stony looking set of professors, and one was really rude to me and my boss, saying, "Why should we collaborate with you? You're going to steal our ideas." That didn't really induce us to try and hand out £1 million, okay? So there is an attitude, but actually I think it's shifting. What I notice is we're incubating 2 companies at the Małopolska Centre of Biotechnology. We've got another company that could spin out, and then there'll be 2 more after that. Different people are more inclined to do it than others. What's really missing is you have to publish for your grants. And before you publish, you have to patent. But often we patent too early. And what's missing is proof of concept grants. Now, some tech transfer office in some universities will provide you a little bit. 50,000 zloty is not enough. You probably need about 200,000 zloty to do something useful, because you've got 12 months to make that patent really strong. And that money isn't there. And I'm going around the various investors here in Poland trying to help the companies find money to take to the next stage, and some potentially very exciting companies. But there are not many investors. They're quite, it may be risk averse. You know, the Americans, they'll just throw you $200,000, $500,000, and they expect one lot to succeed, and they're ready to write the rest off. We don't have the risk taking here. People try to do this research through grants. And my observation of the grant system is it's not very different to the UK, that has the UKRI fund. But if you enter one of these public-private grants, they're very restrictive and they're not agile. And if the research changes, it takes you a year to try and change the theory. And it's just impossible. And it's not conducive to fast research. So Poland is kicking itself in the backside and stopping itself being financially versatile, but the ideas are there, the ability is there. We need training on skill sets, and it isn't the professor usually who will be the really good CEO. It's usually some young hotshot PhD student. It's usually the one that really annoys you. That's usually the person who's got the right phenotype, because there is a CEO phenotype, and we should pick those up. We should train them. There's lots of ways we can train them, trained on the job. There are lots of accelerators around Europe. And for me, it's that proof of concept funding, because you won't get investors until you take it to the next hurdle, but we patent too early. We haven't yet got that next step.
So maybe just a quick comment. So with this understanding, it's clear that where there are companies, like they invest in R&D, so there is this understanding from their side which should go to the academia. So you're saying that there's no trust from academia, you say that in many cases, this is also what Janusz is saying, that in many cases, we don't understand the expectations from the company. And what you're saying about this, this type of the grants, it's later, once there is already some discovery. So what can be done? I mean, and who really should motivate or maybe start this increasing awareness of the scientists, how to talk with the companies, what to expect from the companies, how to understand the companies? So maybe the question, yeah.
I had some meetings with technology transfer centers some time ago, and we discussed this topic, how to improve this situation. Yeah, it's of course all about trust and a little bit also, or maybe not a little bit, also about IP system, but it's also about communication. They told me that there are many projects, many great projects which are conducted parallelly on the business side, parallelly on the academia side, but absolutely independently. So we don't have the points of communication to discuss what is needed. Of course, as Professor Bujnicki said, we need some basic research on the academia side, which are not connected with business issues. But it's about some kind of balance, that we need also this type of basic research projects on the academia side that has this context of commercialization of business issues, meaning giving this technology later to the patients. I think that we absolutely need to create, and I hope that maybe BioMed will be this kind of a platform where universities which are business focused or want to be business focused will have the opportunity to discuss with the business about the needs, about the projects, about potential sources of financing to establish common projects. But it can't be the panel discussion just general. It should be very in-depth discussion, maybe first kind of round table, and then the meetings, talks between each company and each university or institute on the several projects. And I think we absolutely need to start this kind of discussion, and I think we need years, not months, but years to establish better this, but we should start. And I think maybe this discussion would be the right moment to start to create kind of platform to discuss it.
I will give the mic also to Magda, because I just wanted to shed a light from the company side. Well, actually, we have successful collaborations with academia. So one here with MCB. Lately, we have published together even a publication in scientific reports together with MCB. So it is doable, and both sides can achieve here. So it's win-win collaboration. We have also a collaboration with Poznań. It's a grant-based collaboration. So we started with grants to achieve this, but I think that Magda will tell more, because with the Broad Institute, for example, so international collaborations are a little bit different than the Polish ones, because here we are talking now on the Polish perspective, but we have also the international perspective here.
Yeah, I think it looks a bit different from the abroad and in Poland. I spent abroad over 20 years and worked in industry, in academic screening platform as well. So, and I think here you touched two very important things. Janusz was talking about trust, and I don't remember who was talking about communication. I think there are two key elements that need to be somehow improved in Poland. And I think the education is also another one, because when Kaya asked how many of you heard about phenotypic screening, nobody. So it's like educate even students what is drug discovery industry about, because yeah, people just don't know. I learned it working in the industry. So nobody teaches students of biotechnology students how the drug discovery process looks like. So even if they run their own research and maybe something can fit to the pipeline and can be used, they don't know that they should maybe contact the company and start to doing something with this research if they wish, of course. But then it's from the other perspective, the perspective of companies, is just the trust, or this is the perspective of the researcher. They cannot have the attitude that okay, if I share something, that will be stolen and I will have nothing. But this is the way of this is like the task for the companies to ensure them, no, we really want you and you will profit from that, and this is very important what to do. And from the other hand, it's for the companies to understand, and I think big pharmas already understand that this broad academic research and not applied, but just the research can give you sometimes much more than the focused research. Because as also I think Pavel said, that all the big discovery was done by chance. So I think this communication and trust, it's the very important thing. And I think this is what is missing also in Poland, is communication and awareness of what is going on, and the communication, what is going on in industry and academia. So that's my perspective.
And I also would add that we have much more major markets in context of life sciences, like the British one, and there are many best practices which we can implement in Poland. And I absolutely agree that it's also about education, and we need to prepare the future staff for the biotechnology sector, and this is also what BioMed tries to make with the British University, which has the campus here in Wrocław, the Coventry University. We try to establish the new biotechnology faculty, which will be aimed at preparing people which will be business focused with this understanding of the business needs, and which will be focused on the creating technologies, creating drugs or any health solutions for the patients. And I think we shouldn't be afraid of foreign markets. We should take good practices, and we should understand where we are now, because we are still not the major market of life sciences, and we need to learn a lot. And if we have good foreign partners, I think we can do this.
The expression is steal with pride. But from working now for almost 3 years in Poland, you've got some fantastic opportunities and some fantastic ideas. So it's not that they're not there, they are there. It's just learning how to take that next step forward.
So maybe also Professor Bujnicki, from his expertise and collaboration in different European institutions, if you can say what you see that can be quite easily borrowed to our practice, but it's not here yet in Poland, but you know that it's working quite well elsewhere in Europe. I'm not talking about US because it's completely different culture, also in science and industry, but at least maybe somewhere some easy solutions to improve this trust, understanding, and communication.
Well, I think the handling of risk, which has already been mentioned, there are some countries that implemented grant systems where risk, of course, should be managed, but it's allowed. In the Polish grant system, basically, there's no tolerance for risk, and grant agencies require the results of every grant to be published, and it kills science. Of course, on the one hand, it prevents complete nonsense to be funded to some extent, because then it pushes people to be more careful about what they propose. But on the other hand, then if a grant proposal turns nonsense, which sometimes happens, actually, I mean, it never really fully happened to me, but some of my ideas turn out to be wrong. and if I had a grant proposal focused on some idea that would turn to be wrong eventually, I should be allowed to turn this around and turn this failure of testing the idea into something else or return the grant. In Poland, this is not allowed. In many other countries, it is allowed and actually encouraged. So this rigidity and risk aversion is killing the innovations, because science in general is very risky. And when we do science, I mean, there are more failures than successes. Most of our experiments actually fail. Only the few ones are really successful and turned out with the result that we really need. And when we want to turn the research output into something that could be commercialized, then it's way more risky, because there are additional risks beyond purely technical risks. And I'm not going to just enumerate them. But so one thing that we can learn easily, because there are already mechanisms in other countries available, is to change the attitude to risk of the science funding agencies, including the ones that fund basic research as well as the ones that fund applied research. And I can only second the comment about proof of concept grants. They are absolutely essential to make this happen, to go through the valley of death from pure research to innovation.
I think this is a really great direction, because it's clear that the company, they maybe have some buffer for failure, but not big one. And as Janusz has said, the research is mostly about the failing, learning, trying again, failing, running, and so on. And there should be a support which will allow, okay, with the basic grants, we can do this, but then the companies are not involved. If the company is involved in some projects, they rather want to focus for something which will definitely work. I was recently on the commissions for the midterm evaluation of doctoral school, and it was in at least 2 cases where there was a student working with the company that everything was clearly defined, but there were some delays. And our question was, okay, within your research work with the company, are you going a bit broader? Because everything what you do with the company is under IP. You cannot publish it until it's done. What if there will be too large delays? Can you publish something else, like, I don't know, methods development, a research on the side? And then the students was, we haven't thought about this, right? And so as it was said earlier, the research, and of course the company will be interested in this specific part of the research, but still the project can be broader, where academia are happy because we can do something in addition. Company is happy because it's going on that during the direction, but there should be support also from the funding from outside, because it can happen that everything what is around will fail, but then also need to be funding for such approach.
Looking from the perspective of a company, so the easiest and non-risky projects, so services-based projects, are those that we do not need actually the real collaboration with scientists. And if we have the R&D sections where we actually, there is the risk, every high risk, high gain, that's the pharmacy business. So here, if we see the companies dealing with the risk and trying to really find something that's innovative, in Poland with the grants, it's really hard. And you can see that the drug discovery process and the financials that are needed to come to that high gain are really, really huge and cannot be covered with grants. So there are those financial rounds and investors that take part in it. And of course, it's great to have partners in academia that we can test some things. So, for example, we have our idea that it's in silico finding, and we in the collaboration with the academia, we can test it in laboratory, for example. But it should be also on both sides. So, for example, the academia can invent the method for the testing that can be then used, broadly published, and we keep, for example, the rights for the result for a given compound. And it's not that easy actually with that, but it's doable. And also, as Pavel mentioned, with those PhD theses, we have also PhD students in the company. Those are called the applied doctorates. Yes. So actually we do publish things, but those are, you know, the things that are reviews, for example, that we cannot publish something that is the secret of the company, but we can publish the review of the known methodology or publish some other research, and we do that. So it's not, but we talk about it. So with the communication.
Those PhD were not with did you?
Yeah, I know.
I've come across a really interesting model recently. There's a group called the Milner Consortium, and what they've done is about universities and institutes around the Cambridge area. It doesn't have to be like that. And what they've done is companies, and they've all signed the same agreement. I think that might be a bit challenging, but they've all signed the same agreement about confidentiality, about IP and ownership. And there is the concept of free competitive space. We're talking a lot about small molecular weight compounds. The first one you find is almost certainly never ever going to be the drug, all right? So that is not the one you're going to patent. So actually, there isn't any IP to be held. What you can do is always delay publications, but actually that shouldn't stop you publishing. And what the Milner Consortium does is it has roped in 10 companies. It's got the Milner, some investors, and the tech transfer offices work together. And then they put out a call once or twice a year, 1 page. If you can't explain what you're doing in 1 page, you shouldn't be explaining what you're doing. And then they choose to invest, and all 10 companies review this information, and then they select the ones that are most in line with the research they're currently in. And they often fund a postdoc to do the work. And actually, it's working really well. It provides the seed money, it provides the translation, it's pre-agreed, the typical terms. You own the IP you came with, any joint IP is jointly owned. And then there's the first right of refusal. You get some great publications because it's pre-competitive, and it works really well.
It's an interesting concept. Janusz, do you think it might be applied in Poland?
Well, it may be even harder in Poland to have the several companies and institutions agree on such a scheme. In particular, in Poland, still the sharing the IP between the company and between the institutions is a very dangerous field, and institutions don't know how to handle it. And on the other hand, the companies want to have the full control over the IP, individual companies. So I'm not sure if this can work. First, I mean, there needs to be trust, and second, there need to be partners, and third, the legal framework would have to be probably at least audited to see if this is really possible. At this moment, actually, the legal framework is such that the heads of the scientific institutions are responsible for the financial outcome of any such arrangements. So again, it's about the regulations either encouraging taking risks or discouraging. And at this moment, it's very discouraging to take risks. So there would have to be a really serious audit, not just a panel discussion like we're having now, but a very serious audit. What works in Poland and what doesn't, and why? What are the roadblocks and at which level? And and definitely legal roadblocks have to be removed for this to happen. And then of course the mind blocks need to be removed. But this can only happen if we have success stories. If we had in Poland multiple cases where the partner from academia successfully collaborated with the industry and actually received proper revenue from the company that didn't steal the stuff, that would probably change people's minds. And that actually this can happen, that not only collaboration can happen, but the success can happen. And then people would follow the examples of success.
But it's not only about the collaboration between academia and business, but it's also about the spin-offs and spin-outs from universities. For example, in United States, if somebody discovers something or develops something on the university, in quite an easy way, he can take it and just build his spin-off or startup based on this. And in Poland, I had a friend of mine who is a bioinformatic, so I know this story quite well because we talk about this drinking wine. And he told me that he discovered something, doesn't matter on which university, and he tried to establish his spin-off, and the authorities of the universities were absolutely angry on him that he tries to do something like this, and it was just banned on him because of this story. Even though when he finally established this spin-off after 3 or 4 years, he started to look for the business partners, and I recommended him to make the profile on LinkedIn, and he did it, and he wrote assistant professor, and I told him, okay, please write something like business-oriented scientist. The business should know that you are the right person to discuss with. And he told me, no, I can't do this. I asked him why, because university will kill me. So this is the story about the Polish academia side. Of course, there are many places where this is the wind of change, and I hope that this direction would be kept, but still we have a lot to do. But one more thing, because Professor Bujnicki told that we need also some systemic changes, legal changes. And from the June this year, we had something like government plan for development of biomedical sector in Poland, and the medical research agency, which are the right public entity who stands behind this this plan. And I think that maybe this is the job for the medical research agency and for the president of this agency, who is also the governmental representative for biotechnology sector, to start this discussion on the government level, that we need to change also the legal environment to improve and speed up the development of the sector.
I've got one example of good practice, and it blew our minds. So when I was back in business, not in academia, but it's fun changing between the two. I thoroughly recommend it. We were working with Israel, and we wanted to start a project with Israel, but their regulations and law didn't let it. So they went to the government and they changed the law. 2 months later, we started a collaboration, and I thought, wow.
I think this is very nice summary of this discussion, because yes, there are definitely some legal limitations, and definitely there are wind of change from both sides, from the academia and from the industry, and there is a gray zone in between when currently there is very difficult to quickly go to the new, to apply something, to get funding for testing something. It just at the moment it's not existing, and I think this discussion should go further, but unfortunately we are already run out of time, and I guess some people here are getting hungry. So I think we will be continuing this discussion out of the stage, and I hope that with the next such meetings or similar meetings we can go further and probably try to lobby the directions from both ends, academia and industry, because otherwise it will simply not work. Thank you.
Thank you, Janusz. Thank you, Chris, for your great effort to join us here today. And thank you, all of you that are here. If you have any questions, please grab us around here during the lunch. Thank you, Danuta, for a great introduction, and Marta and Magda for the antennas. And of course, Pavel, for
And-
leading that.
And thank you, Kaya, for supporting.